Hello everyone. Today I am going to present an important molecular biology technique called polymerase chain reaction. It's commonly known as PCR. PCR is a powerful laboratory technique used to amplify or make millions of copies of a specific DNA segment. It was developed in 1983 by Carey Mullis and in modern biology and biotechnology. This technique is widely used in medical diagnosis, forensic science, genetic research, and many more fields. The principle of PCR is based on enzymatic application of DNA. In this process, DNA is copied using an enzyme called DNA polymerase enzyme. This enzyme builds a new DNA strand complementary to the template strand. But DNA polymerase cannot start on its own because it needs a primer. Primers means nothing but primers are short DNA sequences that provide a starting point for DNA synthesis. So, overall principle PCR uses template DNA, primers and enzyme to produce multiple DNA copies or million copies of DNA small segments. And next slide shows the components of PCR. In components of PCR, there are 5 components, one DNA template and then second one DNA polymerase and then third one primers, fourth one DNP gears and fifth one buffer system. Now let's look at the important components required for PCR. First one is DNA template, the DNA sample that contains the target gene. Next one DNA polymerase, it's also called as stack polymerase enzyme, a heat stable enzyme that works at high temperatures. Third one is primers, short DNA sequences that bind to the target region. Fourth one is DNTPs, deoxyribonucleotide triphosphates. These are the building blocks of DNA, nothing but ATGC. Adenine, thymine, guanine and then cytosine. At last component is buffer system. It maintains optimal conditions for enzyme activity. It contains magnesium and potassium ions. And then next steps of PCR. PCR consists of 3 major cyclic steps. First one is denaturation, second one is annealing, third one is extension. First one, denaturation. Denaturation means nothing but occurs when the reaction mixture is heated to 94 degrees Celsius for about 15 to 30 minutes. This breaks the hydrogen bonds between the two strands of DNA and converts it into a single stranded DNA. The single strands now act as a template for the production of new strands of DNA. The temperature should be provided for a longer time to ensure the separation of the two strands. Second step, annealing. Annealing means nothing but temperature. The reaction temperature is lowered to 54 to 60 degrees Celsius for around 20 to 40 seconds. The primers bind to their complementary sequences on the template DNA, and then primers or single stranded sequences of DNA or RNA around 20 to 30 bases in the length. They serve as the starting point for the synthesis of DNA. The two separated strands run in the opposite direction and constantly there are two primers, a forward primer and a reverse primer. Last major cyclic step, extension. Extension is also called as elongation. In this step, the temperature is raised to 72 to 80 degrees Celsius. The bases are added to the 3' end of the primer by the Taq polymerase enzyme. It's also called as DNA polymerase enzyme. This elongates the DNA in the 5' to 3' direction. The DNA polymerase adds about 1000 base pair per minute under the optimal condition. Tag polymerase can tolerate very high temperatures. It attaches to the primer and adds DNA bases to the single strand. As a result, a double-stranded DNA molecule is obtained. These three steps are repeated 20 to 40 times in order to obtain a number of sequences of DNA of interest in a very short time period. And now, types of PCR. There are several types of PCR based on applications. First one, real-time PCR. Second one, nested PCR. Third one, multiplex PCR. Fourth one, quantitative PCR. And then fifth one, arbitrary PCR. First, real-time PCR. It's also called as qPCR. In this type, the DNA amplification is detected in real time with the help of a fluorescent reporter. The signal strength of the fluorescent reporter is directly proportional to the number of amplified DNA molecules. Nested PCR, it improves the specificity and reduces errors. This was designed to improve sensitivity and specificity. They reduce the non-specific binding of products due to the amplification of unexpected primer binding sites. Third one, multiplex PCR. This is used for the amplification of multiple targets in a single PCR experiment. It amplifies many different DNA sequences simultaneously. And then fourth one, quantitative PCR. It uses the DNA amplification linearity to detect, characterize and quantify a known sequence in a sample. Fifth one, arbitrary primered PCR. It is a DNA fingerprinting technique based on PCR. It uses primers, the DNA sequence of which is chosen arbitrarily. Next, applications of PCR. PCR has wide applications in different fields. First one, clinical diagnosis. Second one, DNA sequencing. Third one, gene manipulation. Fourth one, comparative genomics. Fifth one, forensic medicine. Sixth one, comparison with gene cloning. First one, clinical diagnosis. The specificity and sensitivity of PCR is highly useful for the diagnosis of various diseases in humans. This include diagnosis of inherited disorders. It's also called as genetic diseases, viral diseases, bacterial diseases, etc. The occurrence of genetic diseases frequently identified by restriction fragment length polymorphism can be employed only when there is a mutation resulting in a detectable change in the length of restriction fragment. Many genetic diseases occur without the involvement of RFLP. RFLP means nothing but restriction fragment length polymorphism. For all such disorders, PCR technique is a real boon as it provides direct information of DNA. This is done by amplification of DNA of the relevant region followed by the direct analysis of PCR products. And then prenatal diagnosis of inherited diseases. PCR is employed in the prenatal diagnosis of inherited diseases by using chorionic villus samples or cells from amniocentesis. Thus, diseases like sickle cell anemia, P. thalassemia and phenylketonuria can be detected by PCR in these samples. Diagnosis of retroviral infections in clinical diagnosis. PCR from cDNA is a valuable tool for diagnosis and monitoring of retroviral infections. Example, HIV infection. Diagnosis of bacterial infections. PCR used for the detection of bacterial infection. Example, tuberculosis. Tuberculosis caused by the bacteria is called as Mycobacterium tuberculosis. Diagnosis of cancer. Several virally induces cancers. Example, cervical cancer caused by human papilloma virus can be detected by using PCR. Further, some cancers will occur due to chromosomal translocation, specifically chromosome 14 and 18 in follicular lymphoma involving known genes are identified by PCR. PCR in sex determination of embryos. Sex of humans and livestock embryos fertilized in vitro can be determined by PCR by using primers and DNA probes specific for sex chromosomes. Further, this technique is also useful to detect sex-linked disorders in fertilized embryos. Second application, DNA sequencing. PCR technique is much simpler and quicker to amplify the DNA. It is covalently used for sequencing. For this purpose, single strands of DNA are required. In asymmetric PCR, pre-prenatal amplification of a single strand is carried out. In another method, strand removal can be achieved by digesting one strand, usually done by exonucleus by its action on 5' phosphorylated strand. Third application, PCR in gene manipulation and expression studies. The advantage with PCR is the primer need not have complementary sequences for the target DNA. Therefore, the sequence of nucleotides in a piece of the gene target DNA can be manipulated and amplified by using PCR. By using this method, coding sequence can be altered to synthesis protein of interest by changing the amino acid sequence. Further, gene manipulations are important in understanding the effects of promoters, initiators, etc. in gene expression studies. PCR is important in the study of mRNAs, the products of gene expression. This is carried out by reverse transcription PCR. Fourth application, PCR in comparative studies of genomes. The differences in the genomes of two different organisms can be measured by PCR with random primers. The products are separated by electrophoresis for comparative identification. PCR is very important in the study evolutionary biology, more specifically referred to as phylogenetic, as a technique which can amplify even minute quantities of DNA from any source, from hair, tissues, bone, or any fossilized material. PCR has revolutionized these studies in paleontology and archaeology. The movie Jurassic Park has created public awareness of the potential applications of PCR. Fifth application, PCR in forensic medicine. A single molecule of DNA from any source, from blood strains, hair, semen, etc. of an individual is adequate for amplification by PCR. Thus, PCR is very important for identification of criminals. The reader may refer DNA fingerprinting technique described elsewhere. Sixth application, a PCR in comparison with gene cloning. PCR has several advantages over the traditional gene cloning techniques. These include better efficiency, minute quantities of starting material of DNA, cost effectiveness, minimal technical skill, time factor, etc. In due course of time, PCR may take over most of the applications of gene cloning. Finally, to conclude, PCR is a revolutionary tool in molecular biology that allows a rapid DNA application. It plays a vital role in medicine, research, forensics, biotechnology. Because of its speed, accuracy, and efficiency, PCR has become one of the most important techniques in modern science and biotechnology. At last, thank you for listening. I hope this presentation helped you understand the principle and applications of PCR clearly.